Mitosis is the process of cell division that results in two genetically identical daughter cells. Here are the steps of mitosis along with brief descriptions for each phase. Interphase. This is the phase before mitosis begins. The cell is actively growing, and its DNA is duplicated to ensure that each daughter cell receives a complete set of genetic information. Prophase. Chromosomes condense and become visible under a microscope. The nuclear envelope breaks down, and the spindle fibers begin to form. The duplicated DNA strands, called chromatids, are now attached at the centromere. Metaphase. Chromosomes align at the cell's equator, known as the metaphase plate. Spindle fibers attach to the centromeres of each chromosome, ensuring proper distribution of genetic material. Anaphase. Centromeres split, and chromatids are pulled apart by the shortening spindle fibers towards opposite poles of the cell. This ensures that each daughter cell will receive an identical set of chromosomes. Telophase. Chromatids, now individual chromosomes, reach the opposite poles of the cell. A new nuclear envelope forms around each set of chromosomes, and the chromosomes begin to decondense. This marks the end of nuclear division. Cytokinesis. In this final stage, the cell's cytoplasm divides into two distinct daughter cells, each with its own nucleus and set of organelles. In animal cells, a cleavage furrow forms, pinching the cell in two, while plant cells form a cell plate between the two daughter cells. These steps ensure the accurate distribution of genetic material to the daughter cells, maintaining the chromosome number and genetic integrity.